Welcome to This Explainer, where we take complex research and break it down into clear concepts so you can get right to the good stuff. Today, we're looking at one of the absolute most profound medical breakthroughs of our lifetime. We are officially moving way past the early days of CRISPR just being this, fascinating laboratory tool. We're looking at its reality today as a genuine approved medical intervention. Okay, let's dive right into this. Here's our roadmap for today. We've got number 1, the first CRISPR cure. Number 2, awakening fetal hemoglobin. Number 3, the human clinical pipeline. Number 4, CRISPR in agriculture. And number 5, milestones and global rules. All right, section 1, the first CRISPR cure. To really appreciate the magnitude of this scientific leap, we kind of have to start by looking at the devastating reality of specific genetic blood diseases. These are conditions that have plagued humanity for generations. But in late 2023, a truly monumental threshold was crossed. A therapy called Casgevy, which was developed by CRISPR Therapeutics and Vertex Pharmaceuticals, was officially approved by the UK's MHRA and the US FDA. This was it. This was the moment decades of basic research completely validated itself. We literally entered the era where we can treat human diseases by rewriting the genome. The two diseases targeted by Casgevy are incredibly severe. On one side, we have sickle cell disease. This is caused by a single tiny point mutation, an E6V substitution, that makes a patient's adult hemoglobin polymerize when oxygen is low. This actually deforms the red blood cells into rigid sickle shapes that physically block blood vessels. It leads to agonizing pain crises and organ damage. Now, on the other side is beta thalassemia. In this condition, mutations severely reduce or just completely abolish the production of beta globin entirely. That causes a severe anemia requiring lifelong blood transfusions. But the core problem here, the real unifying issue, is that both of these diseases severely disrupt adult hemoglobin. Moving right along to section 2, awakening fetal hemoglobin. Now, you might be wondering how we fix that broken adult gene. Well, actually, the solution doesn't involve fixing the broken adult gene at all. Instead, scientists found a brilliant workaround. You see, before we're born, we rely on something called fetal hemoglobin, or HbF, which is excellent at carrying oxygen in the womb. But shortly after birth, a biological switch flips. A transcription factor named BCL11A acts as this master repressor. It essentially binds to our DNA and turns off the fetal hemoglobin factory, allowing the adult hemoglobin to take over. But here's the absolute crucial insight. Fetal hemoglobin doesn't sickle. So researchers realized that if we could just turn back the clock and silence that BCL11A repressor, we could reawaken that perfect fetal hemoglobin and completely bypass the diseased adult hemoglobin. Think about the contrast here between the diseased state and the cured state. In the disease state, before any treatment, those BCL11A levels are high, repressing the gamma globin needed for fetal hemoglobin. The result is low HbF, which causes the red blood cells to sickle, leading to all that pain and organ damage we talked about. But after Casgevy treatment, CRISPR acts like a pair of molecular scissors, specifically disrupting the BCL11A enhancer. Because that repressor is suddenly knocked down, gamma globin is de-repressed. The fetal hemoglobin levels shoot right back up. The result? The cells no longer sickle. In clinical trials, 29 out of 29 sickle cell patients were completely free of pain crises. It's effectively a molecular cure. Just incredible. So how do we actually get this into a patient? Casgevy relies on what we call an ex vivo delivery method. It's essentially a 3-step process. First, hematopoietic stem cells are harvested directly from the patient. Next, outside the body, that's the ex vivo part, these cells are edited using Cas9 RNP delivered via electroporation. That's basically using a tiny electrical pulse to get the CRISPR machinery inside to precisely cut that BCL11A enhancer. And notice, there are no viral vectors involved in this step. Finally, the patient undergoes myeloablative conditioning to clear out their old diseased bone marrow, and their newly edited supercells are infused right back in. The patient literally becomes their own customized cell donor. Let's shift gears to section 3, the human clinical pipeline. Casgevy is massive, but it's really just the tip of the spear. Right now, there are over 50 active CRISPR clinical trials globally, and the delivery methods adapt brilliantly depending on the target. For blindness, like LCA10, a therapy called EDIT-101 uses an AAV5 viral vector injected directly into the subretinal space of the eye to restore correct mRNA splicing. For Duchenne muscular dystrophy, researchers are using an exon skipping strategy. They're literally cutting out a mutation to restore the reading frame and rescue muscle function. And for liver diseases like transthyretin amyloidosis, therapies like Intellia's NTLA-2001 are infused directly into the bloodstream using lipid nanoparticles. These knock out the disease-causing gene right in the liver, showing over a 90% sustained reduction in the toxic protein. And then we have oncology, which represents perhaps the single most ambitious application of CRISPR. Instead of correcting an inherited mutation, scientists are actually engineering immune cells to become smarter, more lethal cancer killers. This brings us to allogeneic CAR T cells. Now, traditional CAR T uses a patient's own cells, which is slow and incredibly expensive. Allogeneic means we can take immune cells from a healthy donor and administer them off the shelf to any cancer patient. But as you can imagine, putting one person's immune cells into another person's body without causing a deadly immune reaction requires some incredibly precise multi-gene edits. CRISPR is used to insert a cancer-hunting CAR transgene directly at the TRAC locus. Simultaneously, CRISPR knocks out the endogenous TRAC to prevent the donor cells from attacking the patient's body. Then it knocks out B2M so the patient's body doesn't reject the donor cells. And finally, it knocks out CD52 so the cells can survive the patient's own chemotherapy. Honestly, it is an absolute masterpiece of cellular engineering. Okay, let's zoom out a bit for Section 4, CRISPR in Agriculture. The power of CRISPR isn't just confined to the hospital clinic. When we look at global ecology, we see this technology actively being used to secure our future food supply. We're talking about editing crops for disease resistance, significant yield improvements, and vital climate resilience so they can withstand harsher global temperatures. It's also transforming animal agriculture. For instance, editing dairy cattle so they're born hornless, which is huge for preventing injuries. We're even seeing nutritional enhancements, like the USDA-approved high oleic soybeans. They knock out a specific gene to create a vastly healthier fatty acid profile, completely free of trans fats. Finally, Section 5, Milestones and Global Rules. As we wrap up this explainer, let's just take a second to look at the timeline of this progress. When you map it out, the speed of innovation is just breathtaking. In 2016, we saw the very first clinical trial in China for lung cancer. Just 3 years later, in 2019, the first U.S. sickle cell patient was treated. In 2020, we achieved the first direct in vivo therapy in the human eye. 2021 gave us the first systemic lipid nanoparticle delivery into the human bloodstream. And then by 2023, less than a decade after those initial trials, we secured the historic full regulatory approval for Casgevy. But while the technology is accelerating, global regulation remains, well, pretty fractured, especially regarding agriculture. The USA regulates on a case-by-case basis, and they often exempt non-transgenic crops, meaning crops that don't contain any foreign DNA. The European Union has historically been the most restrictive, though new regulations have been proposed recently to ease those rules. India takes a fairly progressive stance, treating simple edits as conventional breeding, so they're entirely exempt from GMO regulations. And Japan is among the most permissive globally. There, CRISPR foods simply need to be notified, but not heavily regulated. But while agricultural products might reach the market relatively easily, human therapeutics face a massive hurdle, and that hurdle is cost. The estimated price tag for a single Casgevy treatment is roughly $2.2 million per patient. Which leaves us with a stark reality and a critical question. Diseases like sickle cell disproportionately affect populations in sub-Saharan Africa and India. CRISPR cures now exist, yes, but with multi-million-dollar price tags, we really risk creating a two-tier world where genomic medicine is a luxury only for wealthy nations. The science has succeeded. We know the molecular scissors work. But how do we ensure global health equity and access for all? That is for sure the next great challenge we must solve. Thank you for joining me on this explainer, and as always, keep questioning the world around you.